I want to talk a little bit about what can be an mRNA delivery option in CNS for an in vivo experiment. Um, and that's actually a question from an audience member.
Uh, that maybe I could take that first. So there has been some success with using lipid nanoparticles delivered either intrathecally or using, a pump-based mechanism to deliver the encapsulated messenger RNA. The early generations of lipid nanoparticles, so I mean, and this is going back 15 years or so, again, had a toxicity problem. They had a safety problem. The newer generations of lipid nanoparticles have shown more success in this. And, one of the reasons that it goes back to what I was saying earlier, that local delivery can be an advantage here. So normally a lipid nanoparticle will go, directly to the liver if you deliver it systemically. In the case where you do some version of, again, what I'll, broadly speaking, call local delivery, it actually gives the opportunity to the lipid nanoparticle to deliver to cells that are in proximity to where it is before it goes into circulation, or perhaps doesn't go into circulation at all, and goes to the liver. So again, the site of delivery can be leveraged there as well.
Okay. Um, in the case of using cells as the drug delivery system, how can you guarantee the reproducibility of that protocol?
So I'm not quite sure I understand, but let me just make an assumption and move forward. So if the reproducibility of the protocol refers to the manufacturing, GMP manufacturing of the final product, that's a challenge in cell therapy in general, and RNA cell therapy is not exempt from it. And the way to overcome it is, in essence, be as meticulous as possible in your batch record, and as thorough as possible in training your personnel, and just start to accumulate a body of experience within a given institution. I mean, for us, we built our own GMP manufacturing facility, and has since done, I think, close to 200 GMP manufacturing runs, clinical GMP manufacturing runs in both autologous and allogeneic settings. And I can tell you that the quality of the product that's coming out of our lab today is light years ahead of what was coming out, 4 years ago. So it takes some time to ensure that the quality of the product is significant. You just have to invest in it, because, it's not rocket science, but it's pretty close. It's really Star Warsy kind of stuff. I mean, the notion that you're isolating a cell, growing it, and transfecting it with multiple, RNAs that we've synthesized for the purpose of gaining efficiency and synergy between the combinations, and then, cryopreserving it and sending it off. It's not easy stuff. But it's, if it's effective, which early data certainly suggests it is, then it's certainly worth our investment in time, effort, energy. So it's in some ways it's no different than the kind of in-process controls and quality systems that you would set up with GMP manufacturing any type of therapeutic, whether it's a conventional RNA therapeutic or an RNA cell therapy, or even a small molecule or a biologic. But at another level, I do think that the complexity of making RNA cell therapies is at another level, in terms of the number of steps in a given manufacturing record, and the kind of consistency that you're asking of a cell to take on, as opposed to, say, a small molecule.
Keeping on that manufacturing topic quickly before we do really move on to the audience Q&A. We're already coming up at the end of the hour, believe it or not. You mentioned that, the improvements have been light years in terms of the last 4 years. What and how changed so drastically in those 4 years, and even in the coming short term, because that is one of the topics we want to make sure we get to is, you know, the potential future for the delivery of these therapeutics. What changed in that time, and then what do you see sort of in the foreseeable future from a manufacturing perspective?
I mean, yeah, I can just, you know, start with the cell side. I think, you know, from the conventional nanoparticle-based RNA therapeutic side, I think what changed was the RNA vaccines happened, COVID happened, right? And as a result, that I mean that the amount of investment and interest that went into the field of conventional RNA therapeutics, the reason you have so many in the audience right now listening is in large part attributable to the fact that we're, you know, hopefully at the tail end of the pandemic. So, and have a couple of vaccines that are within this class. From our experience, from the perspective of the RNA cell therapies, you know, I think what the field in general has been looking for is success. So one of the key, you know, question marks in the field has always been, well, what about the half-life of RNA therapies? You know, are these cells, you know, going to survive, and are they going to be, you know, demonstrating a clinical benefit? And so, you know, we wanted to rise to the challenge and move a handful of programs in from sort of preclinical to clinical, and not just into clinical, but, you know, to test our hypotheses in the context of a real almost, you know, a zealous focus on the quality of the product, the sheer scale of generating tens of billions of transfected, healthy, viable, functional cells with RNA, and then the kind of quality package that we put together, we think, sort of gives us the best chance of testing out these hypotheses and seeing some wins in the clinic. Because as soon as you start seeing some wins in the clinic, all of a sudden the entire field of RNA cell therapy becomes really interesting, right? Mm-hmm. So the notion of, so, you know, our RNA vaccines could have gone the other way if the BioNTechs of the world and Modernas of the world had not been so laser focused on the quality and generated a highly effective product that gained approval in record time. If that attempt had resulted in failure, if that readout had resulted in failure, it would be in a deep freeze with conventional RNA vaccines, not to mention its impact on RNA therapeutics. We see that as the same challenge in RNA cell therapy. We want to give ourselves the best shot to see some real meaningful, not just excellent safety, but also real efficacy, and not just a little efficacy. And I think we want to be in a position to just blow every other therapy out of the water. And if we can do that with a select number of indications that I think will be, you know, I think that you'll see RNA cell therapy just explode. Some intriguing data, early data. We want to continue plugging away and really maintain focus on the quality and scale of the product, because that's what's going to give us the best chance of success.
Okay. Thanks, Murat. I want to make sure we get to the audience questions because there are several. Let's talk about regulatory challenges. Walter, perhaps you might be able to comment on this. Do you have any comments on the regulatory challenges associated with mRNA cell therapies?
I'm not a regulatory guy, so I won't spend a lot of time opining on regulatory complexity. What I will say is, a lot of it does go back to the manufacturing that Murat was talking about. That these are complex things that you are making. Even the conventional messenger RNA and a lipid nanoparticle, there are multiple components to that, and a lot of work has gone into making sure that the material is manufactured and then material is formulated that makes it safe, essentially. But beyond that, that's not really my area of expertise.
Yeah.
So, our experience is that most of our experience has been with FDA, and our experience with FDA in general and the Office of Tissue and Advanced Therapies, OTAT in particular, has been incredibly positive. So this is, in essence, a new field, right? I mean, cell therapy in general, again, and RNA cell therapy in particular, has been a very, I mean, we're all sort of learning along the way, and I think the regulators are cognizant of the fact that it would be a tough ask for sponsors to be held to the same level of scrutiny in terms of consistency of the product, the question you raised earlier, with a cell therapy as opposed to, say, a small molecule, right? I mean, there has to be some degree of flexibility, just given the fact that you're working with live human cells, and lot-to-lot variability it's a feature, not a bug of the system, right? And so regulators get that. They're human beings. We've had wonderful dialogues with OTAT. They're thorough. They're very meticulous. They hold us to the highest set of standards, and yet at the same time you can actually have conversations with them. And so, we've submitted 5 INDs over the last 5 years. We're 5 for 5 in INDs over 5 years. All of them have been allowed within the 30-day review period. We've built a little regulatory sort of know-how within our little niche of RNA cell therapy that we find advantageous. And, ultimately, if people are interested in this type of field, I wouldn't be afraid from a regulatory perspective I wouldn't make a regulatory go, no-go decision. I would just lean into it and just prepare for a lot of dialogue with your regulators.
Mm-hmm. Mm-hmm. What are the challenges associated with chronic dosing of mRNA therapeutics?
Immunogenicity.
Yeah, immunogenicity is a big one. Yep. And this is one of the limitations when we start to move beyond a transient expression of a messenger RNA, whether that's a vaccine or a gene editing component, and that is how frequently do you need to dose? And the answer so far appears to be quite frequently. Again, that goes back to something that the circular RNAs may help to address. But intravenous administration of lipid nanoparticle formulated messenger RNA is quite immunogenic, especially when you start to make those subsequent doses beyond the first couple of them.
Yeah, this is a cell is an autologous cell, a cell from a patient that you take out and give back is not immunogenic, right? It can't be, and so we don't worry too much about immunogenicity for our autologous programs. We spend a bit more time thinking about it with our off-the-shelf allo programs, although the type of cell that we use for our allogeneic programs and MSC it's much less class 1 expression, and it's not very immunogenic, although there is some literature suggests it could be in certain indications. We tend to try to choose our indications in ways that are not dependent on long-term lack of immunogenicity. But it's with cell therapy, with RNA cell therapy, it's a much less of an issue relative to the conventional nanoparticle-based RNA therapeutics.
Okay. When you are transfecting the cells with multiple RNA, do you control or target the number of different mRNAs entering each cell?
Yeah. So this is so all of a sudden, when you move from one RNA therapeutic to 2, all of a sudden the questions are, they multiply, right? So what is the ratio? What's the right ratio between these two? What is the cell? What's the type of cell you're going to choose to do the job? And what is the production capacity of that cell? And what's the right ratio? When you move from 2 to 3, as we have for a couple of our programs that are now in the clinic, that question becomes not just a challenge, but a really from a scientific perspective, an incredible opportunity to explore. So all of a sudden you have 3 different ratios that you need to optimize for. We found that you can actually titrate those levels in order to maximize the synergy in terms of the the efficacy and response. So I'll just give you an example with one of our programs, Descartes 25. This is a cancer program that has a bispecific antibody that the mesenchymal stem cell is secreting, along with interleukin 12 and a third RNA that actually we engineer into the membrane in order to target the MSC right to the site of disease. So there are 3 RNAs and therefore an optimal ratio. So the question is, what's the optimal ratio? We found that a given ratio is able to maximize not just the expression, but the synergy between the 2 effector molecules, the bispecific and the interleukin 12, in a way that was just really surprising. So, there's a lot that you can do here when you start talking about the combination of RNA therapeutics. And again, I think a cell as the factory for production and a vehicle for delivery affords a greater opportunity in terms of the number of combinations of RNA therapeutics you can engineer into it, and then you can use the cell to infuse that combination right into the body. So it's a great question, and it's a terrific area of exploration.
Good. Okay. I think we have time for one more audience question, and so it has another regulatory spin to it, but we're going to ask it anyway. Each company has their own proprietary lipid nanoparticle system that may require FDA approval. How do you see the future of upcoming? Ooh, excuse me. Sorry, lost it there for a second. How do you see the future of upcoming different lipid approval just for delivery?
Again, I'm not a regulatory guy, but it is a very good question, and it's one of those. It's going to become a very fundamental one for as we think about those approval processes, not just for each individual company, but for each company as it develops a different product, but using the same delivery technology, where essentially they've literally just swapped out that messenger RNA. It would certainly help things a lot if that could be accelerated, because it was assumed that the delivery piece of it was safe because it is common, and then the messenger RNA. But at this point, again, I'm not a regulatory person. FDA is going to have to make decisions around things like that. It's going to be something that's going to become much more important, but it's a complicated regulatory issue.